Welcome to this lecture on bacterial cell wall. As you know, bacteria is a group of organisms where there may be bacteria, archibacteria, cyanobacteria, then there may be actinomycetes, mycoplasma and rickettsia or chlamydia. So, there may be different types of organism, but in eubacteria only there may be two types, that is the gram-positive bacteria and gram-negative bacteria. And some of the gram-positive bacteria may be having some different character, which is acid-fast bacteria, and there is also a bacteria created without the cell wall, which is called L-form bacteria. We will be starting with eubacteria. We will be noting the location of the cell wall, their structural components and their function. As you can see in this slide, that the bacterial cell wall is the outermost rigid covering of the bacterial cell, but it may be just inside the outer capsule or the glycocalyx layer, and definitely on one side, that is this gram-positive bacterial cell wall is more thick, having a thicker peptidoglycan layer, and this side is little bit thinner with a little bit less amount of peptidoglycan layer. And apart from that, there may be other structural component associated with it. When we go to the next slide, you can see here, this is the basic pattern of the gram-positive bacterial cell wall, where there is almost 85 to 90% constituted by the peptidoglycan, and then there is the cytoplasmic membrane inside. Though there are other layers, we will come to that later on. But the gram-negative cell wall, you can see there is the outermost lipopolysaccharide layer, followed by the peptidoglycan, which is far more thinner, and there is the cytoplasmic membrane inside. So, the bacterial cell wall remains to be the outer protective layer of the bacterial cell. Gram-positive bacterial cell wall is composed of a polysaccharide, which is called N-acetyl muramic acid and N-acetyl glucosamine, or commonly known as NAM and NAG, and they are also having some teichoic acid or lipoteichoic acid or teichouronic acid. Apart from that, there may be the tetrapeptide linkage in between the NAG and NAM, and this particular NAG and NAM are joined by beta 1,4 glycoside linkage, which will actually impart the polysaccharide property to the bacterial cell wall. And you can see here that these are the sugar residue which are interconnected in gram-positive cell wall by different amino acids. These amino acids include the alanine, the glutamic acid, the lysine and the D-alanine. Now here what happens is, in addition to this 4 amino acid, there may be additional glycine linkage present between the 4th and the 3rd amino acid residue of the tetrapeptide, and this particular glycine linkage is called pentaglycine linkage, which is unique for gram-positive bacteria. The teichoic acid can be the wild teichoic acid, can be the lipid teichoic acid. It is actually a derivative of glycerol or ribitol joined by phosphate residue. So, it is a long chain, and as I have told you in the previous slide, that this particular teichoic acid is present all along the cell wall in the peptidoglycan layer. Now, coming to another derivative of teichoic acid, which is the teicuronic acid. This teicuronic acid is having the sugar residue with this 1,6 glycoside linkage, and they also impart stability to the bacterial cell wall. But apart from this particular function, they help in magnesium uptake by the cell wall, and also they help in the binding of the cell wall with the inner membrane in gram-positive bacteria. The S layer is actually helping in the binding of the surface layer with the peptidoglycan, and there are specific SLH domain. You can see here, this SLH domain is not only binding with the cell wall, but it is also binding with the different substrate, like it may bind with the plant cell wall, helping in pathogenicity. So, surface layer is definitely acting as a tool for pathogenic interaction with host plant or host organism. And at the same time, if this surface layer homology domains are absent, then there will be direct interaction between the protein of this layer with the cell wall polymer of the host cell. The next layer is actually the periplasmic space. Now, this periplasmic space, you can see in this gram positive cell wall, hardly there is any position for this periplasmic space. So, it is actually, it is called inner wall zone IWZ, and true periplasmic space is not there in this particular gram positive cell wall. Now, these are more or less the different layers of gram positive bacterial cell wall, where we have discussed the peptidoglycan, we have discussed the periplasmic space and also the surface layer. Gram positive cell wall impart definite shape and rigidity to the bacterial cell. It is a storehouse of different polysaccharide and also different derivatives of those polysaccharide like the teichoic acid, teichouronic acid, etc. And at the same time, it is also having large number of amino acids in the form of tetrapeptide, in the form of pentaglycine, which imparts stability to the bacterial cell. Not only that, it is also protecting the bacteria from the action of different antibiotic, and at the same time there are specific protein which are PBP or penicillin binding protein, which help in the binding of this penicillin with the bacterial cell wall, thereby making them easily prone to that particular antibiotic. And at the same time they will produce some enzyme which actually will destroy those antibiotics like the beta lactamase. So, these are the different functions of gram positive cell wall. You can see here, this is the gram negative bacterial cell wall, where there will be the outer LPS or lipopolysaccharide layer, then there will be the outer periplasmic space followed by the peptidoglycan, and of course when I say peptidoglycan, there will be the surface layer with the porine channel, then there will be inner periplasmic space and also the inner membrane. So, what is important here, that the inner membrane is also considered as a part of the gram negative bacterial cell wall. Now, coming to the different parts of the lipopolysaccharide layer. You can see here that the lipopolysaccharide layer is composed of O-antigen, the core polysaccharide, the disaccharide diphosphate and the fatty acids, which will constitute the lipid. And these are actually definitely bigger molecules, and these components are outside the peptidoglycan layer, which definitely impart some sort of protection to the gram negative bacterial cell wall.
Sir,
yeah.
I have a question. That how O-antigen imparts pathogenicity?
The O-antigen is actually acting as a toxin or endotoxin molecule, which will definitely attack the host cell, will destroy the host cell, will impart some immunogenic reaction, and at the same time it will bring about the end or death of the cell. As you can see here, this particular LPS molecule, though I have said LPS here, it will be O-antigen also, which will be received by high density lipoprotein in human, and also the lipid binding protein of the liver cells will come in contact with that, and that will ultimately bring in the LPS inside the cell, and thereby they will get into the bloodstream, will bind with the neutrophil and the macrophage, and will activate the CD14 cells in the vascular endothelium, and thereby it will spread from one organ to the other organ, and thereby it will bring about a severe toxigenic condition commonly called toxemia, which is a very common cause of death, and we commonly term it as multi-organ failure or multi-organ damage. And when this occurs in gram negative infection, there is no answer, we normally succumb to this type of infection. Now, definitely when I was telling you about the different part of this gram negative bacterial cell wall, I was telling you about the O-antigen. So, I was telling that the O-antigen is acting as an endotoxin. At the same time, it is producing a barrel like protein called omptine, and this omptine is helping as an aspartate protease. It may also act as a pyrogen, inducing fever or infection in host organism. Yes.
I have a question. So, what is the importance of omptin and its role in antimicrobial substance?
You see, omptin, as I told you, is a definite protein which is produced by the LPS layer. Omptin will act as an protease, aspartate protease, which will definitely prevent the aggregation of the tetrapeptide linkage and thereby will prevent microbes. This is one part. At the same time, LPS sometimes inhibit also omptin formation because of this negative aspect. But mind you, at the same time, omptin is also helping in inducing infection. It is a common pyrogen which is not good for you and me. It is causing infection in human and other animals. You can see in this particular slide, there are different tires. The outer tire or O-antigen or the B band is formed of mannose, fucose, and it is N times. So, large number of residues may be attached. Then there is the outer and the inner core of polysaccharide having glucose, rhamnose, galactose, glucose and heptose sugar. And at the same time, there will be the inner core which is formed of heptose and KDO or keto-deoxyoctanoate, and that is followed by a disaccharide residue of NAG which is phosphated, and at the same time the inner fatty acid chain which will constitute the lipid A. So, lipid A actually include the two sugar residue or the disaccharide phosphate along with the long chain fatty acids. This particular residue of lipid A is depending upon the nature of those fatty acids, and these fatty acids are saturated. You can see here it may be varying between 16, 17 and even sometimes 15 or even sometimes 18 residue, and along with this we can also say that it can be triacetylated or tetraacylated or pentaacylated in different types of lipid A molecule. So, this type of lipid A molecule will impart dynamism to the gram negative bacterial lipopolysaccharide. This LPS molecule is a long molecule, and particularly the lipid A is constituted by different length of fatty acid which is imparting dynamism to this particular LPS layer of the gram negative bacterial cell. Obviously, this synthesis of this LPS layer is not a matter of joke. This synthesis will take place in the inner compartment of the bacterial cell wall or the cell membrane, and then gradually they will be transported outward. You can see in this particular figure the O-antigen unit is actually being synthesized by the WZX, WZY and the WZZ molecule, which is placing it just inside the peptidoglycan layer, and this by this time as it is being synthesized, the long residue of this particular O-antigen unit increases, and this O-antigen needs some core lipid A, and this core of this particular lipid A will be synthesized by the MSBA. So, MSBA is producing one part of this molecule, and WZ X, Y and Z is producing the other part, and finally the WAAL is actually connecting these two, and once it is now ready to be launched, it is being launched by this particular group of molecules which are LPT, LPTD, E, A, C, F, G. So, gradually what we have seen here, this particular lipopolysaccharide molecule is being produced in the inner part of the gram negative bacterial cell wall, and then they gradually with the help of some specific carrier protein is being transported to the outer part. It is crossing the peptidoglycan layer and is being fixed in the outer LPS layer. As because LPS is a very large molecule, there will be specific site where it can be modified, and as you can see here, it can be modified by EFTB, it can be modified by CPTA. So, the modification sites can be in the inner lipid A, in the core polysaccharide and also in the outer O-antigen, and this modification will change the particular LPS molecule and will make the bacteria more dynamic.
Sir, I have a question. What is the utility of LPS modification?
Yeah, that is a good question. You see, whenever a particular organism is modifying its own structure, definitely there will be some help for that organism. Say for example, when a particular lipid A is being changed, then the pathogenicity might change. But at the same time, there will be some specific negative aspect for that particular molecule. Say for example, in our liver, when we produce some phosphatase enzyme, that phosphatase will remove the phosphate from the disaccharide residue in the middle part of the lipopolysaccharide layer, and thereby it will render it ineffective. So in defense mechanism also it has got some tremendous role, and at the same time by modifying this particular LPS layer we can reduce the pathogenicity by reducing the toxicity of the toxin molecule. So in one way it is for our own benefit that we have modified the LPS layer, and thereby we have rendered those bacteria ineffective to invade human beings. It is endotoxin production, definitely protection to the gram-negative bacteria, and also it can induce septic shock to the host organism. The next layer is the surface layer or the S layer, which is having unique protein molecules having an approximate size of 25 nanometer, and they are spaced at 2.5 to 3 nanometer. These proteins can be P1, P2 which is oblique, it can be P4 which is square, it can be P6 or hexagonal type of ornamentation. And this particular surface layer will have a unique protein, a cylindrical protein which is called the porine channel. And this porine channel you can see is having 1, 2, 9 different loops, and these loops are formed of different protein molecules. Another unique feature of this particular protein channel is that there is the inner lipophilic layer and the outer is hydrophilic. So the inner molecule is lipophilic and the outer one is hydrophilic. So in one way it will allow both water soluble and fat soluble substances. Rather it will help in the binding of the water soluble substances and thereby will help in the passage of the lipid soluble substances. And not only that, this porine channel will have a surface loop, and this surface loop is having glutamine or aspartic acid residue with calcium ion, which is actually guarding this particular porine channel. So anything and everything is not allowed easily inside the cell membrane, and it will protect the cell membrane. This porine channel will definitely act as a specific structural protein of about 1.5 kDa, and it will have specific hydrophilic and hydrophobic binding area, and at the same time thereby it will balance the different hydrophobicity and hydrophilicity of the cell. It will also help in the entry of the antibiotic molecule inside the gram-negative bacterial cell, because gram-negative bacteria will be targeted by those antibiotics which are mostly inhibiting protein synthesis. The periplasmic space is actually the space in between the outer and the inner layer of the gram-negative cell membrane, and it is housing the peptidoglycan. Obviously it is called outer and the inner periplasmic space. This particular space is actually helping in the transport of nutrients, and at the same time it is preventing the entry of anything and everything inside the bacterial cell, and it also help in the destruction of certain xenobiotic molecules like antibiotics which is trying to enter the bacterial cell. And this particular periplasmic space is followed by, or it is almost housing the peptidoglycan layer. Though in gram-negative bacteria I have said that the peptidoglycan layer is only 15% of the total cell wall composition, but it is also important that this particular peptidoglycan layer, here you can see that this is particularly the gram-positive bacterial cell wall where the peptidoglycan is linked by glycine, but here it is direct, that is having the alanine, glutamate, diaminopimelic acid, alanine, and here also the linkage is between DAP and alanine, and the glycine linkage is not there.
Sir, I have a question.
Yeah.
That does the difference in cell wall structure and components infer any difference in pathogenicity between gram-positive and gram-negative bacteria?
You see, as I told you that the gram-positive bacteria is definitely attacked by some specific antibiotic like penicillin or beta-lactam antibiotic. So naturally the gram-negative bacteria is without those particular sites because peptidoglycan is very little and the cross-linkage is also little. So they are definitely more pathogenic. You cannot really combat the infection of this gram-negative organism. And at the same time, the presence of the LPS layer will increase the virulence of that particular bacteria, making it more pathogenic. The inner membrane of the gram-negative bacterial cell wall is actually a lipoprotein membrane. It is having a unique brown lipoprotein which is having a molecular weight of 7.2 kilodalton, and it is having a lysine residue at the C-terminal end, and there is also a specific hydrophobic colloid or hydrophobic head having the cysteine residue, and this particular lipoprotein is also helping in transport. So what we have learnt is that in a particular bacterial cell wall of gram-negative bacteria, there will be proteins produced in the inner membrane and they will be transported outside. You can see there are two specific core, the outer membrane and the inner membrane, and this inner membrane is having two specific domain, the secretory protein or the sec domain and the trans-activating domain or the trans-activating protein domain, which are both ATP dependent, and they will produce folded exoprotein, and in this folded condition they will be liberated outside and they will be coming out, and whenever they are coming out they are having specific function. These functions include, they may induce pathogenicity acting as a toxin, they may induce entry of specific molecules which are required by the cell, and at the same time they impart dynamism to this particular bacterial cell wall. So what are the functions of this gram-negative bacterial cell wall? They are, number one, they impart dynamism, they act as a toxin, they act as specific receptor, and at the same time they are protecting the organism from the action of different antibiotics, actually sieving the different antibiotic molecules entering the cell and thereby helping in better survivability of that bacterial cell. Now a very pertinent question comes here. Can it be changed? Can you answer that? Can a gram positive be changed to gram negative or vice versa? Yes or no?
Yes.
It can be changed, but the most important thing is that in an experiment, when we change the gram positive bacteria to gram negative bacteria by changing the genome structure, the bacteria will change but not structurally. Structurally it will retain that gram positive properties, though what changes is their nutritional ability. The nutritional ability might change, but the gram characteristics are never changing in a bacteria. What we can say in conclusion, that this particular lecture we have said the different types of gram positive and gram negative bacterial cell wall components. We have discussed their location, we have discussed their function, and we have also discussed that how they impart dynamism to the bacterial cell wall.